Welcome to Chapter 8, Nucleotides and Nucleic Acids. This chapter provides an overview of the chemical nature of the nucleotides and nucleic acids found in most cells. In addition, a more detailed examination of the function of nucleic acids is also presented in this chapter. Key topics include biological function of nucleotides and nucleic acids, structure of common nucleotides, structure of double-stranded DNA, structure of ribonucleic acids, denaturation and annealing of DNA, and last but not least, chemistry of nucleic acids. Let us try and understand the difference between a nucleoside and a nucleotide. But before that, let's look at what a nucleobase is. A nucleobase is a nitrogenous base. For example, pyrimidine or purine. A pyrimidine is a six-membered ring with two nitrogens. And as you can see from the numbering starts with the first nitrogen and it goes along, and numbering of the second nitrogen would be third in this case, and so on and so forth. Purine is a two-ring system with a six-membered and a five-membered ring attached together, and it has four nitrogens. And as you can see, the numbering starts from the N1 and it continues throughout the ring, and then number 7 starts from the nitrogen of the five-membered ring. A nucleoside is a combination of this nitrogenous base plus a pentose ring. A pentose is a five-membered sugar, and usually it's a ribose. And when you combine a purine or a pyrimidine, as shown here, with a pentose ring, you get a nucleoside. A nucleotide is a combination of the nitrogenous base, pentose, and a phosphate. And in this case, a purine or a pyrimidine attached to pentose, which has a phosphate attached to the 5' hydroxyl group. That forms a nucleotide. Now you have to understand the differences between these three molecules. What are the different functions of nucleotides? Nucleotide functions involve energy for metabolism. A classic example is ATP, or adenosine triphosphate. ATP is everywhere inside our body and is a high energy molecule. Enzyme cofactors, for example, NAD+. NAD+ is nicotinamide adenine dinucleotide. It's involved in signal transduction, for example, cyclic adenosine monophosphate. All these are some of the examples of nucleotides, important nucleotides that is in our body. What are the functions of nucleic acids? Nucleic acids are polymers of nucleotides. Nucleic acid functions involve storage of genetic information, for example, DNA. DNA is a nucleic acid. Transmission of genetic information, for example, mRNA or messenger RNA is a nucleic acid. Processing of genetic information, for example, ribozymes. Protein synthesis, which involve tRNA and rRNAs, transfer RNA and ribosomal RNAs. So these are nucleic acids. So these are some of the most important functions. What is the role of nucleic acids in cells? The role of nucleic acids is primarily information storage and transfer inside cells. A gene, as you may know, is a unit of deoxyribonucleic acid, or DNA. It contains information for a functional unit. Genetic information is often communicated via RNA, or ribonucleic acid. The central dogma of molecular biology was first delineated by Francis Crick. Now, this says that the information transfer is unidirectional. Storage of genetic information happens in a DNA. Transfer of genetic information happens in RNA. And translation of genetic information happens via proteins. This slide describes the central dogma of molecular biology. First, DNA, which stores information, gets transcribed onto mRNA. This is a way of information transfer. mRNA is then recognized by ribosomes. Ribosomes are a group of proteins or a protein machinery that converts the information in mRNA and make proteins from it. The way it recognizes mRNA is by using ribosomal RNA. In addition, it also uses transfer RNA, which conjugates amino acids and converts them to polypeptides or proteins, and hence the central dogma of molecular biology. Now that you have seen the big picture, let's go back to the details. The repeating subunit of the nucleic acid polymers, which are DNA and RNA, are nucleotides. Nucleotides, as I have said before, have three components: a pentose ring or a sugar, a phosphate, and a nitrogenous base, which can be either a pyrimidine or a purine. So what are nucleotide bases or nucleobases? The bases are analogous to side chains on amino acids. The bases give each nucleotide the unique characteristics that allow specific interactions. Nucleotides can be divided into two classes based on structure. Purines, for example, adenine and guanine, are purines. There are three different pyrimidines: cytosine, thymine, and uracil. One of the main differences between DNA and RNA is that RNA has uracil instead of thymine, which is present in DNA. Let us take a closer look at purines and pyrimidines. Adenine and guanine are derivatives of purines, whereas cytosine, thymine, and uracil are pyrimidines. Now, if you look at the structural differences, you see that adenine and guanine has 5 nitrogens, 1, 2, 3, 4, 5. And adenine has 1, 2, 3, 4, 5. Purines, especially thymine and uracil, have 2 nitrogens, whereas cytosine has 3 nitrogens. And you can also see that the position of the NH2 in adenine and guanine are different. In addition, these are planar or almost planar structures. They also absorb UV light around 250 to 270 nanometers. Pyrimidine bases. Cytosine is found in both DNA and RNA, whereas thymine is found only in DNA. Uracil is found only in RNA. All are good hydrogen bond donors and acceptors. For example, this NH2 group can either donate 2 hydrogens or accept 1 hydrogen. The carbonyl oxygen can only accept hydrogen bonds. So these are both hydrogen bond donors and acceptors. Cytosine pKa at N3, that is shown here, is about 4.5, whereas thymine at N3 is about 9.5. That's a huge difference. And one of the main reasons why this difference is because this NH is flanked between 2 carbonyls. So it could be either thymine or uracil. This NH has a very high pKa as compared to this nitrogen. Now remember, this nitrogen doesn't have a hydrogen, and that is essentially because at physiological pH, this nitrogen is deprotonated. But if the pH goes below 4.5, this will be protonated, and hence the pKa. Again, neutral molecules at pH 7, these three bases. Adenine and guanine are found in both RNA and DNA. They are purine bases. They are also good hydrogen bond donors and acceptors. As you can see, this NH2, like I said, can be both hydrogen bond donor and acceptor. This nitrogen can be a hydrogen bond acceptor. Adenine N1, the N1 has a pKa of 3.8. Again, similar to pyrimidines. This nitrogen, if the pH goes below 3.8, this nitrogen gets protonated. Guanine pKa at N7. N7 for guanine or adenine is about 2.4, which means if the pH is below 2.4, these nitrogens are protonated. They are neutral molecules at pH 7. So how would you build a nucleoside? Nucleosides have two components. As I said before, a nitrogenous base and a pentose ring. The pentose ring can be beta-D ribofuranose in RNA or beta-2' deoxy-D ribofuranose in DNA. We'll see the difference in a bit. They have different puckered conformations of the sugar ring. So if you look at beta-furanose, and in this case D-ribose, and you know that you can go from an open conformation to a closed conformation, right? This is a Fischer projection to a Haworth projection, and you know how you can go from one to another. A ribose is also called as a furanose, right? Beta-furanose in this case, and beta-D-furanose because this hydroxy is on the right. And the numbering in this case is not 1, 2, 3, but 1', 2', 3', 4', 5'. And hence the numbering 1' to 5'. Now this hydroxy, that is 2' hydroxy, is an essential component. That is because in DNA, this hydroxy is absent. Whereas in RNA, this hydroxy is present. And hence the name ribonucleic acids for RNA and deoxy, because this hydroxy is not there, deoxyribonucleic acid for DNA. This hydroxy in nucleosides is replaced by a base. It could be purine or pyrimidine. And this hydroxy is attached to a phosphate. Ribose ring or a pentose ring is not planar, and there can be 4 possible puckered conformations. The first one is a C2' endo, in which this 2' carbon is above the plane. The second is C2' exo, when the 2' carbon is below the plane and you're comparing it to the plane of the oxygen. In the third case, the C3 carbon could be above the plane of the oxygen, which is C3' endo, or below the plane of the oxygen, which is C3' exo. So these are the 4 possible puckered conformations that are possible for ribose. Now let's talk about the N-Glycosidic bond. In nucleotides, the pentose ring is attached to the nucleobase by an N-Glycosidic bond. So you may remember glycosidic bonds from chapter 6, right? Now we have seen O-glycosidic bonds when a monosaccharide is getting converted to disaccharide or a polysaccharide. Now in this case, it's an N-glycosidic bond because purines or pyrimidines are attached to carbon 1 of ribose. The bond is formed with anomeric carbon of the sugar in beta conformation. The bond is formed to position N1 in pyrimidines, that is N1, and N9 in purines. Please remember this. This bond is quite stable towards hydrolysis, especially in pyrimidines. Bond cleavage, however, is catalyzed by acids. Relative free rotation can occur around the N-glycosidic bond, as shown here. A free rotation of about 0 degrees can happen in synadenosine, where this ring is directly above this sugar. And a rotation of about 180 degrees can result in anti-adenosine, as shown here. An anti-cytidine will look like this. So how do you build a nucleotide? Well, nucleotides have 3 components, as I said before: nitrogenous base, pentose ring, and phosphate. Let's try and understand the phosphate group in nucleotides. Phosphate group is negatively charged at neutral pH. This is really important. Charge is a very important property of a nucleotide and also nucleic acids. Typically attached to 5' position, nucleic acids are built using 5' triphosphates. Usually there are 3 phosphates. This is one, another one attached here, and another one. And because they are 5' triphosphates, they are called as adenosine triphosphate, guanosine triphosphate, thymidine triphosphate, or cytidine triphosphate. Nucleic acids contain 1 phosphate moiety per nucleotide. May be attached to other positions as well. So what is the difference between DNA and RNA? 2 major factors distinguish DNA from RNA. In DNA, the set of bases are ATGC. In RNA, the set of bases are AUGC. Thymine in DNA and uracil in RNA. The second major difference is DNA is deoxyribonucleic acid, which means that the 2' hydroxy in the pentose does not have that hydroxy. The pentose has lost a hydroxy at position 2. Let's compare side by side the structure of adenosine and deoxyadenosine. Adenosine on the left, as you can see, has a hydroxy on 2' position of this ribose, whereas in deoxyadenosine, that hydroxy is replaced by a hydrogen. So let us consider the nomenclature. Ribonucleotides, RNA first. Here you need to know structure, names, and symbols of all these nucleotides, both 1-letter and 3-letter codes as well. For example, adenylate or adenosine 5' monophosphate, or AMP, or A, or adenosine, which is a nucleoside. Nucleotide is guanylate or guanosine 5' monophosphate, G or GMP. Uridylate, which is uridine 5' monophosphate, or UMP, and cytidylate, cytidine 5' monophosphate, or CMP, or cytidine, which is the nucleoside. So as you can see, the difference between a nucleotide name and a nucleoside name is slightly different. And please remember this. For a deoxyribonucleotide, deoxyadenylate or deoxyguanylate, deoxythymidylate or deoxycytidylate. And again, the symbol is DA, where D is small and A is large. So DG, DT, and DC. Again, the nucleoside corresponding to these are deoxyadenosine, deoxyguanosine, deoxythymidine, and deoxycytidine. Besides these nucleotides, there are other nucleotides that are present in cells. And some of the common nucleotides involve ATP, which is triphosphorylated adenine, or cyclic adenosine monophosphate and cyclic guanosine monophosphate, which are important in signaling. Uncommon nucleotides will often have additional groups added to either the sugar or the base. There are lots of uncommon nucleotides in RNA molecules. Nucleic acids are polymers. The linkage between nucleotides is via a phosphodiester bond. Now, for example, starting from the 5' end, this is the nucleobase, and this is the sugar, and this is the nucleotide. Connection between one nucleotide and another nucleotide is via a phosphate diester bond, and this is a covalent bond. This bond is between the 5' phosphate of one nucleotide and the 3' hydroxyl of the other nucleotide. So the nucleotide would be this, and this phosphate from the 5' hydroxy is attached to the 3' hydroxy. The resulting polymer has an alternate phosphate ribose backbone with the nucleotides as side chains. So if you imagine a polypeptide chain in a protein, this ribose and the sugar, the phosphate groups would be the polypeptide backbone, and the bases would be your side chains. You can also represent a nucleic acid in this fashion. It's just a pictorial representation where you do not show the chemical structure of a sugar or a phosphate. Let's look at some properties of these polynucleotides. Covalent bonds formed via phosphodiesters, as you can see, going from 5' to 3' in one segment of this polymer. These are covalently linked to each other. And the backbone is negatively charged. DNA backbone is relatively stable. And one of the major difference between DNA and RNA is DNA is double-stranded. And one chain that is going from 5' to 3' hydrogen bonds to another chain going from 5' to 3'. And this first chain that I described is called as a single-stranded DNA going from 5' to 3'. And this another strand is another single-stranded, and hence the name single-stranded. And when they hydrogen bond, they're called as double-stranded. DNA from mammoths and other long extinct animals could still be obtained and sequenced. The oldest sequenced genome is about 700,000 years from a horse femur. Hydrolysis is accelerated by enzymes, specifically DNases. Remember, when you add SE at the end, it usually represents an enzyme. In contrast, RNA backbone is unstable. As you can see, this is an RNA because it has a 2' hydroxyl. In water, RNA lasts for a few years. In cells, mRNA is usually degraded in few hours. Linear polymers. RNA is linear, and as compared to DNA, they do not form double-stranded RNA between two separate strands. No branching or crosslinks. Directionality, 5' end is different from 3' end. So we can read sequence from 5' to 3'. And that's how you always read DNA sequences or RNA sequences. Now, if you remember how you read protein sequences, N-terminal to a C-terminal end, right? And in case of DNA, you read from what is called as a 5' end to a 3' end. The logic is that the 5' end is because that end has a phosphate from the 5' hydroxy of that ribose sugar or a deoxyribose sugar, and a 3' end is because a 3' hydroxy in an RNA or a DNA is free. Hydrogen bonding interactions exist between nucleotides. Specifically, two bases can hydrogen bond to form a base pair. For monomers, large number of base pairs are possible. In polynucleotides, only few possibilities exist. They're called as Watson-Crick base pairs, and they predominate in double-stranded DNA. For example, A pairs with T and C pairs with G, hence AT, CG. Please remember this. Purine pairs with pyrimidine. An example of Watson-Crick hydrogen bonding is shown in this slide. If you consider a double-stranded DNA as shown here, there are two different hydrogen bonding interactions. Adenine hydrogen bonding to thymine, or guanine hydrogen bonding to cytosine. Let's look at the first example. In adenine, the nitrogen NH2 on C6 carbon donates a hydrogen bond to the carbonyl oxygen in thymine. The NH in thymine donates a hydrogen bond to the nitrogen, which is the N1 in adenine. Now, as you can see, there are two hydrogen bonds in adenine and thymine. When you compare that to cytosine and guanine, you can see that there are three hydrogen bonds that exist between these two pairs. And as a result, the hydrogen bonding interactions between guanine and cytosine is stronger than adenine-thymine pair. Bases affect the properties of nucleotides and nucleic acids. Conjugated bonds in nucleotide bases result in high absorbance of UV light. We use absorbance of 260 nanometers to quantitate DNA concentration. The bases are hydrophobic and aromatic. Aromatic interactions between bases help align them. Hydrogen bonding between bases and different nucleic acids is the basis of the structure of DNA.